Hello, we're Saja, and this is our presentation over water quality part 2. So, as we learned in the last lab, many different factors go into water quality, and in order to better understand these factors, we will observe the different levels of TDS, conductivity, ammonium, alkalinity, and pH within different samples.
So a little overview of what exactly we did in this lab. The first experiment was ICP-OES. We prepared 10 milliliters of a stock solution, used that stock solution to measure and plot some absorbance, and then established a calibration curve based on our data. The next one was an IC calibration, which this was partly done by the TAs. We calibrated a solution we prepared a solution with 7 anion solution, used the IC measuring instrument and measured the intensity of each ion, and then plotted this data. We also analyzed the stability to ensure it stayed constant. And then preparing the sample, we put 1.5 milliliters of the diluted sample into a vial. The third experiment was a TOC, also partly done by the TAs for calibration. You had to add 32.9 milliliters of low TOC water to a standard vial and add about 0.4 milliliters of stock solution to the vial. Next, we added 0.4 milliliters of the concentrated phosphoric acid to the vial and put a cap on. And then to prepare the sample, we added 39.6 milliliters of the sample to the vials, and then acidified the samples with 0.4 milliliters of phosphoric acid and also capped those again.
All right, for our data, we're going to start off with the monovalent ICPs and OES. So for this, we have the 4 different intensities that were shown through the concentrations. We can see over here how each group had a different concentration aside from the TA, which are recorded, and then you have the intensity and the absorbance levels. Right, and then here we have some graphs showing kind of the intensity absorbance versus the concentration for the potassium ones. They're both kind of got that linear increase as intensity increases, so does the concentration. Right, and here is the sodium ones. Once again, pretty similar. All right, and then on the same page, we have a little bit more calculations. So by using the slopes and the y-intercepts of the last slides of our graphs, with the given intensity values, we can calculate the concentration. And down here we have some of the averages and then the standard deviations of both the potassium and the sodium. For this, we can both see that the bay water or the lake water have very, very, very high concentrations compared to all of the other ones. Next, we have the divalent ICP OES, kind of similar to our last one. Each group had a different concentration, and there were different intensities that were shown through, and the recorded absorbance levels are shown also on the graph. All right, here's our graphs. They're also linear, kind of just like the first part of our lab, with the just going up. Same thing here for calcium. Right, and then similarly, using the slope and the y-intercept of the previous graphs with given intensities, we were able to calculate the concentrations. And from there, we had to use the dilution factor for the sea waters or the bay water because it was simply too high for the machine to read, so we had to dilute it for it to read. All of the highlighted values over here that are kind of pinkish, those came out as negatives, and so I cast them as outliers and did not include them in the averages. Over here we have the averages. Next, we have the IC and your cation concentrations for each of the water samples. Here is kind of the bar graph showing the concentrations of chloride, which is in the blue, and sulfate, which is in the orange, I believe. I'm sorry, I said that backward. Chloride is actually in the blue and sulfate is in the orange. But you can see that they're all extremely low, at least from here, compared to the bay water, which are very, very high levels. Here's a little bit closer where I took out the bay water so we could see everything on a little bit closer level. Over here we have the diluted bay water, which is a little bit different. And so you can kind of see all that. All right, next we have the salinity and the TOCs. Each of these were given to us, and then the salinity was calculated. So yeah, for this, they're all roughly kind of around the same values, with bay water being a little bit higher than the tap water and the Lake Austin water. And the salinity with the raw bay water being the highest and the treated bay water being the lowest. Here is another, just kind of showing the alkalinity and the pH, all that good stuff. Yeah. Here we have our TDS values, the averages. Number 5 here is going to be our micropores. 4 is the treated bay. 3 is the Lake Austin raw. Number 2 is the bay raw water, and number 1 is going to be your raw water. Once again, bay raw water is going to be the highest, and it looks like treated bay water is the lowest for the TDS in parts per million. Here we have the major cations for each of the different cations in the sample water. So for bay water, you can see that they're a lot higher than the other ones for just about all of the cations. Here is a bar graph representing the TDS values and the average for each of the water samples, which in the gray is the electroconductivity of the TDS in parts per million. In the orange is the IC and the ICP and alkalinity TDS, while in the blue is going to be your evaporation dish TDS. So once again, raw bay water is significantly higher than all of the other ones. And then we have electroneutrality. Once again, raw bay water has much, much higher compared to all the other ones, which we can see that as they're treated, it goes down. Down here is going to be our percent error, which was calculated using the electroneutrality values. And yeah.
So for the data analysis, I'm just going to be reiterating the data that Julia just explained to you. And so starting with the monovalent ICP-OES, as concentration increases, absorbance values also increase. So basically we see a direct relationship between the intensity and the concentration. And the way that we could improve the precision and accuracy of these experiments would go into calibration of the equipment that we're using, standard operating procedures to make sure that we're taking things at correct time intervals. So that goes into steady data collection, because we have to make sure that everything is happening consistently. And then also environmental controls to make sure that everything that we're testing is in the same temperature, same humidity, and everything is just stable. For divalent ICP-OES, the results are also reasonable for our samples. For example, magnesium and calcium showed us that the intensity of the emitted radiation is directly proportional to the concentration of the analyte. For TOC, all of the TOC values are relatively similar, with the raw waters holding a slightly higher concentration than that of the others. To discuss the units that we used, mgc per L refers to the amount of carbon present in milligrams per liter of solution. So that quantifies the concentration of organic carbon compounds in our aqueous solutions, such as dissolved organic carbon in water samples. For carbonate, to talk about alkalinity versus the total dissolved solvents and electroneutrality, alkalinity is associated with the amount of bicarbonate and carbonate ions in the water sample. High alkalinity contributes to higher TDS and electroneutrality values, and cations and ions composition affect the electroneutrality. Cations will add positive charges, and anions will add negative charges to said sample. And the alkalinity equation can be simplified to consider just the concentration of whatever species we're talking about, like the alkalinity contributing species. The concentration of that is how we can also figure out the alkalinity.
With our TDS section, one aspect of the data that was of interest was having lower TDS for the raw lake water than the microsample. This procedure could have been could have been made more efficient by using a heating plate instead of just leaving out the plate to dry to reduce the possibility of dust or other air particles from entering the dish. And another area of interest was the ICP and the IC having varying values due to small differences in volume changing their reading. And similarly, that can also affect the electroneutrality data. Other aspects that we should include within this data is temperature and pressure, because they can also affect electroneutrality. And for our calculation, H plus and OH minus may be removed to simplify the calculation.
To conclude everything we've learned for interpreting our data, we learned that water quality is important because contaminants can be harmful, lead to lots of various health issues, and we've been learning that over the years through engineering. The data presents the collected experimental data regarding certain qualities of water to determine the condition of water samples. Is it safe to drink? Assessment of our experimental error, some things to consider was the calibration of the measurement tools. It's important as it can affect the accuracy and the precision of the data collected. So it's possible there could have been some sort of experimental error if we did not calibrate correctly, as well as the process of heating the TDS samples on the evaporation disk. Sometimes heating and evaporating too much can make the TDS values left behind not as accurate. How do we relate this to environmental engineering? Well, water system design and management, and basically how measuring how safe water is to drink is a very large part of environmental engineering. It's one of the biggest fields you can go into. So it's important that we learn and understand the chemical and analytical knowledge behind water quality as we begin our major here. So there's a lot of expanding research on water quality, and we should understand the basics as the field grows. So thank you for listening to our presentation. Here's our citations.